All right, I want to talk about your tissues lab today. So before I go over our long PowerPoint, I want to mention that there's a couple other things that we're going to be looking at today. Since this is mostly all microscope slides, I've got a couple ways for you to look at them online. So the first one is a virtual scope. And this one, you have a virtual scope lab that you can look at, and you can read about a couple of different types of tissues. And if you go through here, you need to put in this username and password. And if you did the last mitosis lab, you should have already used this for the onion root tip and the whitefish blastula. But we're going to be looking at a couple more slides this time. 8 different ones, I think. There's a comprehensive activity guide that tells you each slide to look at. And there's also a corresponding video description. You should definitely watch those. Now, what I like is that looking at these real slides, either on a microscope or on this virtual way, shows you that you're not just going to have a slide with like one type of tissue on it. Usually all of your organs and your tissues and your membranes are made of multiple types of tissue. So on different parts of the slide, you might have fat some places on the slide, or you might have areolar connective tissue here, or you might have cartilage here, or bone, or skin and muscle, all different areas. So each slide is not just going to consist of one specific type of tissue, it's going to consist of a bunch. So this sort of introductory one is going to show you how we've got lots of different types of tissues, and they're not necessarily going to be just muscle or just epithelial tissue or just connective tissue. They might contain multiple different types of tissues. APR is going to be really good to look at just one specific type of tissue. APR doesn't have the whole slide, it just has like one view of the slide, and that view of the slide is like the perfect view of like that perfect piece of tissue that you want to be able to label and look at all the different things. So that will be good to look at because APR labels everything for you and lets you look at all the different areas. This is really good. There's a virtual histology slide box. And this is for cells and tissues right here, so you can look at all the different epithelial cells. It kind of tells you about epithelium, and you can look at all of these here. This, again, is really good because this has the whole entire organ or the entire membrane. So this is like a bile duct, so it shows you all the different areas. So some of it is going to be epithelial tissue, right? But some of it's going to be connective tissue under there, and it's really nice because you can move around in this whole slide. There's blood vessels in there. So we have epithelial tissue as well as connective tissue. So you can kind of look at these slides. It gets a little bit more confusing if they're not labeled. So that's why I'd say maybe start with APR and start with the V-scope that kind of goes through, and you can watch the videos with everything and then look at these later. But you can see there's blood vessels that are lined with simple squamous epithelium here, and bile ducts here, and we've got lots of this here is simple cuboidal epithelium. So there's lots of good stuff, but you've also got all this connective tissue in the middle. So you can see in real slides, we don't just have one type of tissue, we've got lots of different types of tissues. And histology is really complicated. If you ever take any other histology courses, you'll find that out. So there's lots of good slides here you can practice looking at. This tissues playlist talks all about different tissues. I have 2 tissues crash courses here, mostly about connective tissues because I think those are one of the more complicated ones to look at. In lecture, we'll talk a little bit more about the intro basic tissues as well as epithelial tissues. And let's see, the other thing that I have for you guys is right here. There's a bunch of practice. Now, this PowerPoint is not something for you to print out because it's really long. It's a lot of slides, but if you download the PowerPoint and open it, it's definitely going to be useful to you. And along with that, I have this tissue chart. And so I have a Word document too, so you guys can just type in it. This lists all the tissues, and you can use this to organize your notes. Write about the location of that tissue, the function of it, and what structures or features it is. How many cell layers does it have, or what types of tissue, like what types of fibers are in it, what types of cells are in it, and things like that. Right, so the Histology Lab practice PowerPoint, you guys should really use it. It's really useful. As I said, also use APR, don't forget that. There's a lot of good study materials that you guys have. So this is basically showing you, it is, as you can see, it is really long. if you go into presentation mode, you can see that here is the epithelium over here, and this is what you need to identify, and these are the different types of epithelium that you'll need to identify. All right, so then if we go to the next slide, you'll say, okay, this, you don't get the whole slide, but this is like the good area of tissue that you have to identify. And then if you go to the next part, it says, okay, this is the basement membrane. This is the nuclei. This is the squamous shape. This is stratified squamous non-keratinized epithelium. This is a description of it. Go through. This is another stratified squamous non-keratinized epithelium. Here's another. So they don't all look exactly the same. So I would practice looking at these. Right, this is like 4 different versions. Now this one is a new one. This is keratinized stratified squamous epithelium. And so it has lots, like it's very, another instructor made this, but it's very well done. And so this goes through all the different types of tissues that you need to know, and it labels all of them, and it's a really good practice. And you get to see that not all of them look the same. The nice thing about it is APR has one example of each. Your book has one example of each. And sometimes it's the same as APR, and it'll be similar to the ones that you see in the PowerPoint. I don't want you to think that every single type of tissue looks exactly like that. So this PowerPoint shows you these are both, they look a little different, but they're both simple cuboidal epithelium. This is simple columnar, 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 this one has microvilli and goblet cells in it. Ooh, this one has really pretty cilia up here. This one's pseudostratified columnar. So we've got lots of these different ones to look at. So I'm going to give you on the lab quiz, you'll have to answer some of the questions from that V-scope activity, and then I'm going to give you a slide and ask you to identify what type of tissue it is, including any cell surface modifications if present. So in this case, you would have to say that it's pseudostratified epithelium with cilia, right? Pseudostratified columnar epithelium with cilia, or ciliated pseudostratified columnar epithelium. So you need to be able to tell me that, right? This is ciliated pseudostratified columnar. So I said with cell surface modifications, and I'll mention what that is when we get there. So just go through, this goes through connective tissue. What do you need to identify? All the different types of connective tissue. Make sure you look at this one. It's really useful. Then if you keep going, there's a lot. We go through bone, cartilage, different types of cartilage, all the different types of muscle, and then finally nervous tissue. And at the end, you have the summary slides where it shows you these are all cardiac muscle, skeletal muscle, smooth muscle. This is all the blood. And it just basically shows you all of them without the labels, well, most of them, in one slide. These are the bones. This is all the stratified squamous non-keratinized, so you can see them all together. So these are the summary slides at the end. So as I said before, this will be best viewed on your device. You don't need to print them out, but it's probably one of your best study aids for this chapter. All right, so now that I've talked through everything else you should be looking through, I do want to go through the lab slides, of which there are a lot of them, unfortunately. I am not going to read every slide to you about each type of tissue because you guys can do that on your own, but I will try to talk about the most important parts. So this is chapter 5, tissues. Histology is the study of tissues, and tissues are groups of similar cells as well as extracellular material that performs a common function. We have 4 types of tissue. We're going to be focusing mostly on epithelial and connective tissue in this class because muscle tissue will cover extensively in chapter 10 and nervous tissue will cover extensively in chapter 12, but I do still want you to be able to identify muscle and nervous tissue under the microscope. So we'll first talk about epithelial tissue, and it is composed of 1 or more layers of closely packed cells. These cells contain little to no extracellular matrix. Extracellular matrix means things that are out, like other things that are not the cell, outside stuff. It's non-living, it's not the cell, it's like proteins and things. It covers the body's surfaces and it lines the body's cavities. It also forms the majority of glands, which we'll talk more about in lecture. As we said, it's mostly very, very tightly packed cells, and these cells they have a polarity. So they're going to have an apical surface, which is the surface that is exposed to the external environment. So it's right here. This is the apical surface. They have a lateral surface, which is where the 2 epithelial cells are connected next to each other, and they have a basal surface, which is where the epithelial cells are connected to the basement membrane or the cell below them, depending if there's multiple layers. The attachment to the basement membrane is important. The basement membrane is the barrier between the epithelium and the connective tissue. So when we talk about membranes in lecture, we'll talk about how most of these epithelial tissues are sitting right on top of a connective tissue, and the basement membrane is that barrier in between them. And in some areas, the connective tissue that underlies it is going to be called the lamina propria. So if you see lamina propria in APR, it's just the connective tissue that underlines this epithelial tissue. This is another example. This blood vessel is lined by simple squamous epithelium. So this is the lumen. The inside of the blood vessel is called the lumen. Think about the inside of a straw that you drink through. And so this would be the apical surface of this cell here that opens towards the lumen. And then this would be the basal surface that sits towards the connective tissue that is underneath it. Epithelial tissues lack blood vessels, right? So if you've ever gotten a paper cut on your finger and it didn't bleed, it was just through that outer layer of the epidermis. And so you don't have any blood vessels there. The nutrients are either obtained across the apical surface or from the basal surface. So depending on where it is, like right here, this blood vessel, obviously it's getting nutrients just directly from the blood across the apical surface. Here, it would get nutrients from the underlying connective tissue across the basement membrane. These are also usually highly innervated, so they have lots of nerves and sensors, and then they have a good capacity for regeneration. We'll talk a little bit about tissue repair in the integumentary chapter. So with our epithelial cells, especially our skin cells and things like that, if we get a scrape, we can just grow new skin. So they can undergo mitosis very readily. So some of the functions of epithelial tissue would be that it physically protects you, it's selectively permeable, it allows passages of some substances but not others. Some of them are specialized to secrete things, and then we have a lot of specialized epithelium that can actually feel and sense sensations, and we'll talk more about that in chapter 16. All right, so now let's see, how do we identify these under the microscope? So all of that was interesting, but we really want to know how to identify these under the microscope. That's what we're doing in this lab. So how do we name epithelial tissues? So epithelial tissues are named and classified by a couple of things. One of them is the number of cell layers. So if it's one cell layer thick and all cells come in contact with the basement membrane, it is called simple epithelium. If it is two or more layers thick and only the basal layer is in contact with the basement membrane, it is called a stratified epithelium. If it looks all jumbled and it's kind of in between, if it looks stratified but it is really not, it appears layered but all cells come in contact with the basement membrane, they may not all reach the apical surface, it's called a pseudo-stratified or falsely stratified epithelium. So when we name epithelial tissues, we always say simple, stratified, or pseudo-stratified first. So the number of layers is going to be the first part of the name. So simple epithelium are one cell layer thick, stratified epithelium are more than one cell layer thick. The next thing we classify them by is the size or the shape of the cell and the shape of the cell on the apical surface. So the basal surface, they're almost always like this cuboidal or low columnar, but the apical surface is where we look at the shape of the cell. So we name the tissue based on the shape of the cell at the apical surface. All right, and also pseudostratified is pretty much always columnar. Or for this class, it will definitely always be columnar. So now we're going to classify them by cell shape, and this will be the second part of the name. So squamous cells are like flat and wide and irregular. They look a little bit like a fried egg. Cuboidal cells are cubes, right? They're as tall as they are wide. The nucleus is very spherical and in the center of the cell, whereas the squamous cell, the nucleus is flat and elongated in a like wide lengthwise direction. Columnar cells are slender and tall, and the nucleus is oval, but it's taller than it is wide. And then transitional cells are a type of cells that change in shape depending on the stretch of the epithelium. And these are mostly found in the bladder and the ureters, right? And they're going to change in shape whether you have a full or an empty bladder. So these transitional cells here, you can see that they look, they're almost always stratified, but they look more thickly stratified if the bladder is empty. If the bladder is thin, they look more a single layer, and that sometimes they look Squamous when the bladder is distended and they look, you know, I don't know, they look very rounded and dome shaped when the bladder is empty. And so for this one, you don't call it stratified, you just call it transitional epithelium. It's kind of the exception to the rule. So if we were going to name something, right, the three names would be the number of layers first, then the shape of the cell second. So 1 number of layers, 2 shape of the cell, and 3 epithelium. It's usually a three names. So we would say, like, it could be a simple squamous epithelium or something like that. The other thing we'll look at is something called a cell surface modification. And this is going to be a modification that happens at the apical surface of the cell, right? So the top of the cell, or like that top layer here. And so there's a couple of different ones. Now the first one I'll talk to you is about keratinization. One thing to know is that keratinization only happens in stratified squamous epithelium. So stratified squamous epithelium can either be keratinized, which is your skin. That's like the example. That is what it is, the only place it is. Or it can be non-keratinized, like in the esophagus. So we have a lot of layers of these egg-shaped cells, and at the top they look really flat. Keratinization is when these cells fill up with keratin. And when they fill up with keratin, they die and they lose their nucleus. And so the top part usually stains kind of a dark color, and it looks really, really flaky. And in some areas, like especially in thick skin, it's really thick. And so it helps waterproof and protect your skin. These next three, cilia, goblet cells, and microvilli, are only going to be in columnar epithelium. So either simple or stratified columnar, or simple or pseudostratified columnar epithelium are pretty common to have these cell surface modifications. So in this picture, we see these like clear looking cells here. These are called goblet cells. And we can see them here. These are called goblet cells. So goblet cells are in all these. Goblet cells are these little single-celled glands or unicellular glands that secrete this mucin, which is a component of mucus. So whenever we have a mucous membrane, right, lining our digestive tract, lining our respiratory tract, we're going to have these goblet cells because we need to have mucus on top of our mucous membranes. The other thing we have are these extensions of the plasma membrane called cilia or microvilli. Which ones are longer? We learned about this in our cell structures lab. So the microvilli are tiny and they don't move on their own. They're going to increase the surface area for absorption. We call this the brush border because it looks like, if you've ever felt velvet, you probably felt it. You can feel the individual little like bristles or like a really soft brush, but you can't really tell. You can't really see them because they're so small and close together. So that's this fuzzy edge that you can't really tell. This is the brush border of or microvilli. And then we have cilia. So if you can look and you can sort of see that it's long and you can see the individual strands, you know that's cilia. Cilia actually move under their own power and they beat and they're going to move the mucus along the surface of the epithelium. Or like in the oviducts, they're going to move the egg from the ovary into the uterus. So cilia, goblet cells, and microvilli are going to be in columnar epithelium, whereas keratinization is going to be found only in stratified squamous epithelium. So we have a bunch of different types of epithelium. So simple versus stratified. Simple means there's one layer, stratified means there's two or more. Pseudostratified is a type of simple because it's falsely stratified. It just looks like it has many layers. Any of these that have this star on them, it means they have ciliated and non-ciliated subtypes. And the non-ciliated subtypes usually have microvilli. Stratified squamous is once again the only one that can be keratinized. So, I'm going to flip through these slides here. I want you guys to read them and look at them, but it's going to take me way too long to like just read these slides to you. You probably don't want to hear it either. So we have simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium, and that exists in a non-ciliated or ciliated forms. The non-ciliated, once again, has microvilli, which is that brush border. It also has goblet cells. The ciliated simple columnar is going to have cilia and goblet cells. And the nice thing is it tells you where they're located. For this lab, you don't need to memorize where they're located, but you can add that to the location in your little chart I told you guys to write out, and it will be important to know for lecture. Now, this is part of chapter 5, so it's not going to be on your next exam, on the first exam. It'll be on the second exam. So don't get too caught up on that yet. Pseudostratified ciliated. Epithelium is pseudostratified columnar epithelium can also be ciliated or non-ciliated, right? And they've got goblet cells. Let's see. Stratified squamous epithelium, right, can be keratinized or non-keratinized. Stratified cuboidal, stratified columnar, both of these are more rare. And then finally, transitional epithelium is found in the bladder. This right here is like a decision chart. This is how you can look at a tissue and try to figure out which type of epithelium cells it is. So it's kind of like a dichotomous key. You can go through and like answer yes or no to each of these to try to figure out which cell it is. You also can make your own dichotomous key in the lab manual. Next, we're going to move on to connective tissue. So connective tissue is, there's a lot of different types of connective tissue. It's the most abundant and widely distributed tissue in the body. It contains blood, bone, fat, ligaments, not muscle. Anything that's not muscle, nerve, or epithelial tissue is connective tissue. Connective tissues all share three basic components: cells, protein fibers, and ground substance. So we know what the cells are, and these cells are not nearly as tightly packed. They're not going to be like sitting and attached to their neighbors like they were in the epithelial tissue. The protein fibers and the ground substance, we didn't have very much of that before in epithelial tissue, but we have a lot of it in our connective tissue. Protein fibers and ground substance together are called the extracellular matrix because they're outside of the cell. We have two different types of cells that are found. Resident cells always live in the connective tissue. Wandering cells kind of wander through, and usually those are immune cells. So we're not going to focus on that much now because you don't know about the immune system yet, but we'll learn about it in 169. So these resident cells maintain and support and repair the extracellular matrix, so the ground substance and the protein fibers. A really common type, especially in the connective tissue proper, is a fibroblast. These are flat cells with tapered ends. They produce fibers, especially collagen, and ground substance. Adipocytes are also known as fat cells, and they appear in small clusters in different types of connective tissues. But if it's all, like pretty much if adipose cells or fat cells predominate, we call that adipose connective tissue. Mesenchymal cells are embryonic stem cells, or I guess they're adult stem cells that came from the embryo eventually. They divide to replace damaged cells. One replaces the mesenchymal cell and the other becomes a committed cell, like it might become a skin cell or something like that. And then finally we have a couple of immune cells that live in these connective tissues. They're called fixed macrophages. They're stationed there. They're like the police officer that's stationed in a different area. I'm not going to ask you to identify those yet until we get to the immune cells. As we said again, wandering cells are going to be types of white blood cells that move through the connective tissue. We're not going to focus too much on that in the lab. All right, then those were cells. There's also some specialized types of cells, and once we get to like the bone and the cartilage, they have special names, and I'll mention those. Now we have connective tissues. Next, they have protein fibers. So there's three different types of fibers to know. We have collagen fibers. Collagen I think of as like rope. It is flexible and it is strong, but it is not stretchy. So they are going to be found a lot in tendons and ligaments. Reticular fibers are kind of similar to collagen, but they're thinner and they kind of branch a lot, and they form the framework of some of your like meaty organs like the lymph nodes and the spleen and the liver and things like that. And then finally we have elastic fiber, and as the name implies, they contain the protein elastin and they stretch and recoil easily. They're found in the skin and the walls of those large arteries that have to absorb the force of the heartbeat. So here, the collagen is actually this, this picture shows both elastic fibers and collagen fibers. So this pink part, the light pink part that's hard to see, is actually the thicker elastic fiber, the thicker collagen fiber, and this thin one is elastic. I will mention that all of these tissues here that you can see are stained with different stains. Your book doesn't really go into this, and it can be kind of confusing because the different fibers are going to stain different colors based on different things. And I'm actually going to really quickly take, because I forgot to talk to you about this, I'm going to take a moment and go over different types of stains because I think it will help you understand why all the tissues look a little bit different. All right, so if we go to this virtual histology slide area, I think that there is one, let's see, types of tissues. cannot remember. There's an area that talks about different types of stains. I think it's in this website somewhere. All right, so different stains can be used to visualize each type of fiber in connective tissues. And so this is pretty important. So we've talked about the different types of fibers, right? So we had the collagen fibers, the elastic fibers, and the reticular fibers. And they're going to look different in all the different things, and they'll look different in all the different specimens depending on which type of stains we use. And so we can look for the, let's say this silver stain right here. So this silver stain is the stain that will show the reticular fibers. And so whenever you see a picture of like reticular connective tissue and it looks kind of black like this, and it looks like these black little branching guys, that's because it was stained with silver stain. And we have, in actual lab, we have one lymph node that's stained with silver stain, but we have other lymph nodes that are not stained with silver stain. And the other lymph nodes look like you can't see these reticular fibers well unless you use this special silver stain. So this silver stain will let you see the reticular fibers and they'll look black like this. But other stains, you won't see them as well. So we have to use different stains to visualize different fibers. All right, so that was the silver stain can be used to see the reticular fibers. Now if we look here, we can see this is a blood vessel here. And we have a couple of different types of stains. So here, if you look right here, H&E is a very common type of stain. So hematoxylin and eosin. And let me move my little, let me move this. I know you can't see all of this, but I will read it to you. So collagen in H&E is stained pink or red. And muscle is stained pink, so they can be a little bit confusing, but elastic fibers and reticular fibers are colorless, so you won't see it. So this one is going to be an H&E stain of this blood vessel. And so we can see the collagen fibers here. We can see elastic fibers, but like you can't really see them very well. They kind of look clear. They are colorless. And then muscle, you can see it, but it doesn't look very different. It's also kind of colorless. So now if we change to a different stain, this is the same thing. We're going to change to the azon stain. This one is going to make collagen look blue. And so we can see here that collagen is blue, whereas the other ones, so this is collagen, the outer wall is mostly collagen. Elastic fibers are like this sort of clear colorless, and then reticular fibers are unstained. And muscle here is going to be stained kind of a red or a pink. And so now we can differentiate between muscle and collagen fibers because sometimes they look similar. Now this Verhoff stain is the last stain, and this Verhoff stain is pretty cool because elastic fibers stain black. So any of those, like the elastic connective tissue or the elastic cartilage, you will see a lot of times these like really darkly staining black elastic fibers, and that's because they're stained with this special Verhoff stain. And so, and sometimes you'll see collagen stained blue, and sometimes you'll see it stained pink. And that's because they're, might be it's H&E or azon or Verhoff. It uses all these different stains. And then whenever you want to see these reticular fibers, if you see them as black, it's going to be that silver stain. And so here, this is cool because you've got the collagen is stained black, or excuse me, collagen is stained red and elastic is stained black. So you can see the collagen again here is red, and then these elastic fibers are going to be black. Otherwise, you can't differentiate them. That well. So you also don't memorize something by its color because these different stains, right? Like if you memorize, oh, collagen's pink, well, sometimes it's blue or sometimes it's red, and so on. So different stains can make each tissue look a little bit different. So I just wanted to mention that before we start going into the connective tissue because all these different fibers look different with these different stains. All right, so that was a quick aside talking about different types of stains for all these different slides. So if we look at this slide here, you can see that the elastic fibers are all going to be stained this really dark color, and they collagen fibers are stained this pink or red color. And so this must be like that Verhoff, which is going to be showing you really well those elastic fibers. And this one right here, you see that reticular fibers are actually stained black, so this must be the silver stain. I think it's a little bit of a failing of the book that it doesn't really tell you which stain each of the tissues was stained with, but this isn't actually a histology class, so it's probably not that important. But if you do take a histology class later, the different stains will be important to know. At least know about them. All right, so let us keep going and talking about different types of connective tissue. All right, now we talked about the cell types, we talked about the protein fibers, now we have to talk about the ground substance. So the ground substance is non-cellular material produced by the connective tissue cells that's other than the fibers. It can either be viscous, like the blood has, like the plasma would be the ground substance of the blood. It can be semi-solid in the case of cartilage, or it can be solid in the case of bone. Ground substance plus the protein fibers together are often called the extracellular matrix, or abbreviated as the ECM. So we can see here we have our extracellular matrix, which is made of our ground substance and our protein fibers, and then we have our different cells. All right, so as we look through, we can classify connective tissue in different ways. We'll start with a connective tissue proper. This does a lot of sort of binding and padding and things like that. Then we have our supportive connective tissue, which is cartilage and bone. And then finally, we have our fluid connective tissues, which are blood and lymph. Lymph does not really have any cellular components, so we're not going to look at it under the microscope, but we will look at blood. Spongy bone we'll talk more about in the bone chapter, but we're not going to focus on as much today. So loose connective tissue contains areolar, adipose, and reticular. And this, if you look at it, it looks kind of looser. It has a higher percentage of ground substance and a lower percentage of fibers, and fewer cells also. And so it looks a lot less cluttered. So areolar right here, it contains both elastic and collagen fibers. So those pictures up here, like this, is going to be a good areolar connective tissue. And so it's going to contain a bunch of fibroblasts. All of these connective tissue proper are going to contain lots of fibroblasts, although the adipose is going to contain mostly adipose, but all the rest of them contain lots of fibroblasts. So most of these are fibroblasts here, then you have collagen and elastic fibers. And you can see how there's a lot of white space, so it's loose. This one also has a lot of white space, or it looks like white space at least. It's mostly these fat cells, and so this is, the nucleus is pushed off to the side and it's filled with these triglycerides, right, the storage form of fats. Next we have reticular connective tissue. Remember, this one is, if we see the reticular fibers, it's been stained with a silver stain. And usually, a lot of times it will have white blood cells in it because it makes up the spleen that has a lot of white blood cells. It makes up parts of the liver that has a lot of white blood cells. It makes up the lymph nodes, which has a lot of white blood cells. And so those are the leukocytes. Leukocyte is the fancy term for white blood cell. It also makes up the bone marrow, which has lots of blood cells, the thymus. All of those were loose. You could see lots of white space. Now we have the dense connective tissue proper. Dense regular connective tissue, dense irregular connective tissue, and elastic connective tissue. This has a lot of protein fibers, mostly collagen. So here we can see all of these collagen fibers. All of this pink is collagen, not muscle, it's collagen. Here, this is going to be the nucleus of the fibroblast. The fibroblast made all these collagen. In dense regular, all the collagen fibers go in the same direction. They all go parallel to each other. But they have very few blood vessels. Most of the other connective tissue has a good amount of blood supply. This one here, you can see there's also collagen bundles, but instead of going in all parallel lines, they kind of mix up and they all go in different directions. This is going to be similar except for the direction of the collagen fibers. Then finally we have elastic connective tissue, and as you can see, this, if it's staining black, it's the Verhoff stain again, and so you can see that there's a lot of elastic connective tissue here. It can be hard to tell if there's elastic tissue in there unless you use this special stain though, because remember, it doesn't, with the normal H&E stain, it doesn't always stain. So elastic connective tissue has a lot of elastic fibers in it, so like the trachea, the vocal cords, large arteries, things that need to be able to recoil. All right, so this is a little sort of dichotomous key or flow chart that lets you try to identify the different types of connective tissue proper. So you can look at that to help you identify. All right, now let's move on to supporting connective tissue. So two different types are cartilage and bone. So we'll start with cartilage first. Cartilage is has a semi-solid extracellular matrix. It's not as hard as bone. It's kind of gel-like. It has collagen, it should say collagen, and elastic protein fibers in it. The cells are called chondrocytes. So chondro for cartilage and cyte for cell. Those are the mature cells. And they occupy small spaces called lacunae, or they live in them. They're their little houses. And most of them, most types, are surrounded by a dense, irregular connective tissue covering called the perichondrium. So peri means around, and chondro is cartilage, right? So it surrounds the cartilage. It's more flexible than bone, but it does not actually, in mature cartilage, it does not have any blood vessels. We'll go over these again in the skeletal system because we'll talk a lot about cartilage in the skeletal system. So the three types that we'll talk about and that you need to identify are hyaline cartilage, fibrocartilage, and elastic cartilage. And this is a nice little cartoon that I like that kind of compares them. Hyaline cartilage, you cannot see any of the protein fibers. They don't stain, they're not visible. So it looks very clear. The chondrocytes, to me, look a little bit like eyeballs or bullseyes. So you can see like the circle here is the lacunae, and then the inner circle is the cell, and then the dark part in the middle is going to be the nucleus of the cell. So it looks a little bit like eyeballs to me. So if you see these little eyeballs or the bullseyes, think chondrocytes and think cartilage. So here we have got, you can't see any stain fibers that stain. This is going to be collagen that's pink. It's probably like an H&E stain here. And this one here is going to be elastic fibers. And the elastic fibers are pink in this one and not black. So I think it's the H&E and not the Virchow stain. But a lot of them will show up black because black is really easy to see those elastic fibers. So if you see black, to me, it looks almost like paint splatters, the elastic cartilage, where you've got this like splattery look in here, like short little a lot of branching elastic fibers that look almost like paint smears or paint splatters. Whereas the fibrocartilage, the collagen, it's like wavy collagen that all goes together, and it pushes the chondrocytes sort of into these parallel bundles. You can see these little parallel bundles of chondrocytes. All right, so once again, hyaline cartilage, glassy appearance. You don't see any fibers. You can see the chondrocytes, but no fibers. Fibrocartilage, this is that azon stain, right, where we said that collagen, it looks blue. It has these wavy collagen fibers right here. And it kind of pushes these chondrocytes into these parallel lines because the collagen sort of pushes them off to the side. And then finally, elastic cartilage. As I said, it has these sort of short branching elastic fibers, and they look a little bit like sort of splattery and smeary to me. And that's going to be found like in your external ear, right? If you sleep on it like this, it's going to bounce back, as well as the epiglottis. So bone is going to be another supporting connective tissue. The bone cells, instead of being called chondrocytes for cartilage, are called osteocytes for bone. Osteo means bone, but they are also housed within those spaces called lacunae. It is not as obvious in bone, but we're actually going to go through the structure of bone in detail in chapter 7. So I'm going to briefly talk about it here, but we're going to learn more about it in chapter 7. Two types are compact and spongy bone. Compact bone is going to be very distinct. You will know it when you see it. And it has these cylindrical osteons. It looks a little bit like growth rings in a tree. And so this is the compact bone right here. So this looks like, this is the central canal, and then we have these concentric layers of bone called the concentric lamellae. And these little black lines right here are the osteocytes. So to me, it looks a little bit like growth rings in a tree. So this one should just scream compact bone. This one's easy to identify. You guys should be able to get this one. We're going to talk about all these different labels and all these different like pieces of bone when we get to chapter 7. So don't worry about memorizing all these labels for this next exam, or the next lab quiz this week, just be able to identify it. All right, so this is a little sort of flow chart for looking at the supporting connective tissues and trying to figure out which one is which. So you can use this to help you figure that out. Now last we have the fluid connective tissues. So blood is the only one we're really looking at because lymph doesn't have any cellular components, so it's pretty clear. We don't need to look at that under the microscope as much. But blood is a lot more interesting. So it's a fluid connective tissue with formed elements. Erythrocytes are red blood cells, leukocytes are white blood cells, and then platelets or thrombocytes help the blood clot. The liquid ground substance is called plasma, and it contains dissolved proteins. So here we can see that these like pinkish cells with a central pallor, which means they have a white spot in the middle, are these erythrocytes. That's the red blood cells. The ones here that are kind of speckled and purple, these are the white blood cells or leukocytes. And then these tiny little fragments over there are platelets. And then the white stuff in the back is just the plasma, which is the ground substance. All right, that concluded connective tissue. I know this is a lot of different tissues. This lab is probably one of the harder labs for everyone to wrap their minds around, but stay with me. We're almost done. We only have nervous and muscle tissue left. There's only 3 types of muscle tissue and 1 type of nervous tissue, so we're almost there. We're also going to be focusing on muscle tissue in chapter 10 and nervous tissue in chapter 12, so I'm going to keep this pretty brief. I do want you to be able to identify these types of tissues under the microscope, but you don't need to know all the details until we get to chapters 10 and 12. So muscle tissue is important because it contracts when stimulated. We have 3 different types: skeletal, cardiac, and smooth. So I like this little cartoon. It kind of compares them just like it did with cartilage. So skeletal muscles are very big. They're very long. They have multiple nuclei that are pushed off to the side, and they look striped. We call these striations. Cardiac muscles are also striped. They also have striations, but they're a lot smaller and they branch. And they have 1 to 2 nuclei per cell. And they have these special little cell-to-cell junctions called intercalated discs. And they are going to contain both gap junctions and desmosomes, which we'll talk about more in lecture. And so you can see the striations, but they're not quite as big. They don't take up like the whole slide like the skeletal muscles do. You can see they're branching and they have those special intercalated discs. And then finally, smooth muscle does not have the stripes. It's not striated. It has 1 nuclei per cell, and so the nuclei are fairly evenly distributed throughout the muscle. Whereas in dense regular connective tissue, which it can be easy to mistake it for, you'll have big areas that don't have any nuclei. So skeletal muscle, once again, this is skeletal muscle. It's striated, it's very large, it has the nuclei pushed off to the side. Cardiac muscle is striated. It has these dark little intercalated discs. The cells are a little bit smaller, and they each only have 1 to 2 nuclei, and they branch. And then finally, smooth muscle, each cell has 1 nuclei, so the nuclei are evenly distributed throughout. They're sort of tapered, and they do not have any striations. So here's our flow chart for looking at different types of muscles and figuring out which is which. All right, nervous tissue is our last tissue. I'm so excited. And it's very obvious if it's nervous tissue. Nervous tissue is located in the brain and the spinal cord and the nerves. The big cells are called neurons. They receive, transmit, and process nerve impulses. They look like spiders. So this, like these would be the dendrites, and then this would be the axon. It looks like a big spider-shaped cell. Then we have a lot of little tiny glial cells that kind of support and surround the neuron. So this is what it looks like. These big guys here are these neurons. They're the big spider-like cells, and these tiny little guys here are the glial cells kind of surrounding him. And that is it. I know there's a lot of slides, but it's just a lot of information for you to look through and fill out that chart. Please ask me questions. This one can get a little bit confusing. You will have, once again, 3 attempts for the lab quiz. The first, I think, 9 questions are going to be from the vScope virtual lab, so make sure you look at that and you can use that, have it in front of you while you do the quiz. So that'll be good. Okay, let me know if you have any questions.